Hi, I'm Brian Ceronie from the BartsMS group in London, and we're interested in the mechanism of action of Cladribine. So we know that Cladribine, like AM2017, works really well in MS, and we know from the Clarity and the KRMS trials that, like AM2017, Cladribine is able to suppress B cells by almost 80%. But unlike AM2017, it doesn't cause such a substantial reduction in T cells. And so we know that Cladribine is more selective for the B cell population than AM2017, and we think this might be a key driver behind its efficacy. So what we've been trying to understand is why it might be more selective for B cells and whether there's specific populations of B cells that Cladribine is better for. So at Barts, we developed a compassionate use program of Cladribine for people who are otherwise ineligible for other therapies. And we wanted to use this data that we've collected to understand whether there's specific types of B cells that Cladribine is more effective against. And so we did a cross-sectional study involving 40 participants, comparing 8 people on Cladribine just prior to their next dose compared to AM2017 and compared to both healthy and non-MS controls. And we analyzed this data using flow cytometry, looking at specific subsets of B cells. And what we found that just prior to the 44-week before the next dose of Cladribine, we found that memory B cells in particular were significantly suppressed throughout the entire course of treatment for Cladribine, which was similar to AM2017 and was very different from the healthy and non-MS controls. And when we looked at the specific subsets of memory B cells, we also found that both switched and unswitched memory B cells, as well as the interleukin-2 expressing B cells, were significantly depleted by Cladribine just prior to their next dose, suggesting a long-term and sustained memory B cell depletion by the drug, which we think is key in underlying its effects. And we wanted to try and understand a bit more about why Cladribine might work in this way. So we looked a bit at the gene expression data of lymphocytes and of germ cells in the blood and in lymphoid tissues. And what we found using publicly available data is that the most important thing to understand the mechanism of action of Cladribine is the ratio of the enzymes that break down or phosphorylate Cladribine to make it toxic, specifically the adenosine deaminase and the deoxycytidine kinase enzyme, and also the 5 nucleotidases. If you look at these ratios, you find that the highest ratios of all these enzymes are in the B cell compartment, which suggests why Cladribine might be a selective B cell inhibitor, and it might be suppressing the specific subsets of the B cell compartment while not leading to the same reconstitution and secondary autoimmune phenomena that you get with AM2017. So our future directions for this project is to try and clinically correlate this with a bigger cohort of patients to show that the clinical effects of Cladribine are driven by memory B cell depletion. And we'd also like to look a bit more at certain subsets of the memory B cells, in particular the B regulatory cells, to see how this might influence the B cell compartment as a whole.